we eat a cholesterol-containing food, in the liver and intestine, the cholesterol metabolizes into primary and secondary bile acids. Several studies have shown there is a relation between these bile acids and neurodegenerative diseases like Alzheimer's and Parkinson. This relation raises the question of which bile acids are exactly involved in the process of degeneration. Answering this question became difficult when we encountered with bile acids that we could not easily differentiate them. It is because they have same molecular formula and molecular weight, but they have very small differences in atomic arrangements. So what do we do now? The solution is to use traveling wave ion mobility spectrometry with electrospray ionization source. This analytical technique permits the separation of ions based on collision cross-section differences. To separate these isomeric molecules, we try different set of strategies. 1. We fragmented away the identical functional group of molecules to form smaller ions and augment the differences. As a result, we got to have different arrival time for each ion. 2. We enhanced the collision cross-section differences by add-up ion formation, which worked pretty well for these enantiomeric isomers. Also, the sodium salt add-up formation promoted the separation of these stereoisomeric ions, even when we injected a mixture of them. 3. We took advantage of ions' tendency to form clusters. Formed dimers modified the gas phase behavior of ions in a way to have unique arrival time. We could even separate them in a mixture solution. So, it seems really nice. We could distinguish conformationally similar bile acids to further investigate their correlation with different neurodegenerative diseases. But wait a minute, we still haven't answered a philosophical question. What is exactly happening? To shine light on this question, we performed theoretical calculations with density functional theory and compared them to our experimental results. Do you remember we could separate these stereoisomers? We found out that alpha and beta isomers tend to form planar and bent ring conformations. These conformational change, along with sodium localization, enhance the collision cross-section differences. That's why we could see separation. For the isomeric molecules, the position of sodium differed greatly for each isomer, which resulted in the formation of ions with varying degrees of compactness. Consequently, isomers showed unique arrival time. By means of DFT, we could identify similar trends for other molecules and explain their gas phase behavior. So, now we know how we can separate these challenging isomeric molecules, and also we know why we can separate them. With these findings, we have taken one more step towards biomarker detection, which is of particular importance for further drug discovery studies. My name is Poster11 Hadavi, PhD candidate at Amphora Institute, Maastricht University.